So we are on to the last topic about photosynthesis. And again, just to refresh our learning target at the end of this lesson is that you guys should be able to describe the major features and chemical events in photosynthesis and respiration, and also explain the importance of chlorophyll and other pigments in photosynthesis. So, a plant placed in a dark closet, they literally starve. Without light, the light cannot generate ATP or NADPH. And without these two critical energy and electron carriers, the plant cannot feed itself. Once its stored reserves are gone, the plant dies. The plant's life thus depends on the light reactions of photosynthesis, which occur in the membranes of the chloroplasts. So, we have already seen the pigments and proteins of the chloroplast's thylakoid membranes are organized into photosystems. More specifically, the thylakoid membranes contain two types of photosystems. We have photosystem 1 and we also have photosystem 2. An electron transport chain connects the two photosystems. So just a quick recall on your previous lessons about electron transport chain. So electron transport chain is a group of proteins that shuttle electrons from carrier to carrier, releasing energy with each step. So as you will see, the electron transport chain that links the photosystem 1 and photosystem 2 provides energy for ATP synthesis. A second electron transport chain extending from photosystem 1 ends in the production of NADPH. Okay, so first we will be discussing about the photosystem 1. So remember, the first important step is when light strikes. So photosynthesis begins in the cluster of pigment molecules of photosystem 2. These pigments absorb light and transfer the energy to a chlorophyll A reaction center, where it boosts two electrons to an orbital with a higher energy level. The excited electrons, now packed with potential energy, are ejected from the reaction center chlorophyll A molecule and grabbed by the first protein in the electron transfer chain that links the two photosystems, as you can see in the picture. Now the question is, how does the reaction center chlorophyll A molecule replace these two electrons? So the answer would be in water. So they come from water. So remember, the chemical formula for water would be H2O. So water, which donates two electrons when it splits into oxygen gas and two protons, which is H+. So chlorophyll A picks up the electrons. The protons are released into the thylakoid space, and oxygen is either used in the plant's respiration or released in the environment. Meanwhile, the chloroplast uses the potential energy in the electrons to create a proton gradient, as you can see here in the picture. So as the electrons pass along the electron transport chain, the energy they lose drives the active transport of protons from the stroma to the thylakoid space. The resulting proton gradient across the thylakoid membrane represents a form of potential energy. So as you can see here in the part where it is boxed, see here, the arrows go down, which signifies that the energy, as it is being transported from one carrier to another, the energy goes down. And as I mentioned earlier, the energy that is lost during that process drives the active transport of protons, which are originally placed in the or found in the stroma, so that they will be transported into the thylakoid space or into the thylakoid lumen. Next, we have ATP synthase. So ATP synthase is the enzyme complex that transforms the gradient's potential energy into chemical energy in the form of ATP. A channel in the ATP synthase allows protons trapped inside the thylakoid space to return to the chloroplast's stroma. As the gradient dissipates, energy is released. Now, the ATP synthase enzyme uses this released energy to add phosphate to ADP, or adenosine diphosphate. And when it does that, it generates ATP, or adenosine triphosphate. So this is how you would imagine the ATP synthase would look like. So from ADP, it transforms that to ATP. Now, this mechanism is similar to using a dam to produce electricity. You see, in a dam, water accumulating behind represents potential energy, like the proton gradient across the thylakoid membrane. To harness this potential energy, the dam's operators allow water to gush through a large pipe at the dam's base, turning blades that spins an electric generator. Likewise, ATP synthase generates ATP as it accumulates, or as it allows accumulated protons to pass from the thylakoid space into the lumen. Next, we have, or from the thylakoid space into the stroma rather. Next, we have electrons from photosystem 1 reduce NADP plus to NADPH. Now, photosystem 1 functions much as photosystem 2 does, which means we again will be needing light. So photon energy strikes energy-absorbing antenna pigment molecules, which pass the energy to the reaction center chlorophyll A. The reactive chlorophyll molecule ejects electrons to an electron carrier molecule in a second electron transport chain. The boosted electrons in photosystem 1 are then placed with electron passing down the first electron transport chain from photosystem 2. And that is what you can see here in the picture. Now, the second electron transport chain passes the electrons to a molecule of NADP+, as you can see here, forming NADPH. This NADPH is the electron carrier that will reduce carbon dioxide in the carbon reactions, while ATP will provide the energy. Next, we have the carbon reactions that produce carbohydrates. Now, the carbon reactions are also called as the Calvin cycle, and it occurs in the stroma. The Calvin cycle is the metabolic pathway that uses NADPH and ATP to assemble carbon dioxide molecules into 3-carbon carbohydrate molecules. These products are eventually assembled into glucose and other sugars. So as you can see here, this part is the light-independent reaction, or the carbon reactions, or also known as the Calvin cycle. And the main product for that would be glucose and other sugars. So the first step of the Calvin cycle is known as the carbon fixation. So the initial incorporation of carbon from carbon dioxide into an organic compound is what we call as the carbon fixation stage. Specifically, carbon dioxide combines with ribulose bisphosphate, or RUBP, which is a 5-carbon sugar with 2 phosphate groups. So take note, ribulose bisphosphate, or RUBP, it has 5 carbon and 2 phosphate groups. Now, Rubisco is the enzyme that catalyzes this first reaction, as also you can see here in this part. Now, the 6-carbon product of the initial reaction immediately breaks down into 3 carbon molecules, which we call as PGA. So further, Our further steps convert PGA to PGAL, which is the carbohydrate product that leaves the Calvin cycle. So PGA stands for phosphoglyceric acid, while PGAL stands for phosphoglyceraldehyde. So that is the next step. Now, for the second major step, we have the reduction phase. Now, in this phase, PGA is converted to G3P with the use of ATP and NADPH. So, NADPH donates its electrons to a 3-carbon intermediate for it to become G3P. So, G3P stands for glyceraldehyde 3-phosphate. Now, after the reduction phase, we have the carbohydrate formation. In this step, G3P is now turned into carbohydrates such as sugar. On the last step, we have the regeneration phase. In the regeneration phase, the G3P is recycled to regenerate RUBP to make the process continue. And take note as well that this process would require ATP. So what happens is that ATP would act on G3P for it to make another RUBP. And that RUBP produced in the regeneration phase will be again used to start the cycle. Now, in general, a G3P molecule contains 3 fixed carbon atoms, so it takes two molecules of G3Ps to build a 6-carbon glucose molecule. And if we're going to be counting everything, so it would take 6 turns of the cycle, which means we will be needing 6 molecules of carbon dioxide, 18 molecules of ATP, and 12 molecules of NADPH for us to produce 1 glucose. So several fates await the carbohydrates produced in the carbon reactions. A plant cell uses about half of the sugar as fuel for their own cellular respiration. So this is a metabolic pathway that we will be discussing in the future. Roots, flowers, fruits, seeds, and other non-photosynthetic plant parts could not grow without sugar shipments from green leaves and stems. Plants also combine glucose with other substances to manufacture additional compounds, including amino acid and a host of economically important products such as rubber, medicines, and spices. Moreover, glucose molecules are the building blocks of the cellulose wall that surrounds every plant cell. Wood is mostly made of cellulose. The timber in the world's forest therefore stores enormous amounts of carbon. So do vast deposits of coal and other fossil fuels, which are the remains of plants and other organisms that lived long ago. Burning wood or fossil fuels releases this stored carbon into the atmosphere as carbon dioxide. As the amount of carbon dioxide in the atmosphere has increased, Earth's average temperature has risen. Now, if a plant produces more glucose than it immediately needs for respiration or building cell walls, it may store the excess as starch. Carbohydrate-rich tubers and grains such as potatoes, rice, corn, and wheat are all energy-storing plant organs. Some plants, including sugarcane and sugar beets, store energy as sucrose instead. Table sugar comes from these crops, and in addition, people use starch from corn kernels and sugar from sugarcane to produce biofuels such as ethanol.